Okay, so now we're going to go into the GOGAT cycle. So Dr. Thomas did say he doesn't expect you guys to draw the entire thing, but I'm going to go over the most three important steps of the reaction, starting with sucrose. Also, just to limit time, I'm not going to draw the structures, I'll just draw the names. But actually, before we go into the steps, let's go over what the GOGAT cycle is. So the GOGAT cycle, all it is the shuffling of amine groups. So you're just moving them around, really. So just know how to draw them and understanding how each step is working. So you have sucrose coming in, and it makes oxaloacetate. Okay. And then through aspartate, which is the enzyme that we're using for this specific step, we make aspartate. And then we use asparagine synthetase, which is denoted as AS. And in this step, we use ATP, which makes AMP, plus pyrophosphate, and that makes asparagine. So this is one pathway from the GOGAT cycle that's really important. Another one is glutamate, which in the presence of NH4 and the input of energy, along with glutamine synthetase, which I'm going to denote as GS, it can make glutamine. But also, I noticed I spelled glutamate wrong. But also, glutamate can also go through another pathway in which it can make alpha ketoglutarate. And this one requires NADP+, which gets reduced to NADPH, and the release of NH4. And in this pathway, use the enzyme glutamate dehydrogenase, which I'm going to denote as GDH. Okay? And then alpha ketoglutarate can be converted into glutamate. So we can actually go back to glutamate. However, it requires the GOGAT enzyme, which is the glutamate synthase. And in this one, you use NADPH, and you make NADP+. So notice how to go from glutamate to alpha ketoglutarate, we use NADP+, right? So that means that glutamate requires NADP+. So it only makes sense for us to make NADP+ when we're trying to get back to glutamate. And this is the GOGAT enzyme, also denoted as glutamate synthase. Okay, so familiarize yourself with this pathway. I know it can seem a little overwhelming, but once you break it down like this, it makes a lot more sense. Another thing I wanted to go into is it's really important to know how these amino acids and structures are related to one another. So alpha ketoglutarate has multiple types of derivatives, and these are the derivatives that make alpha ketoglutarate, and these are the derivatives that make oxaloacetate. So just familiarizing and recognizing the types of amino acids that can make these different types of structures. Also, right, you have pyruvate. This was like the first midterm, right? So alanine can make pyruvate because there's similar structures, so just know that. Also, we also have how some amino acids can be made by simple transamination reactions. I thought I'd show you guys this slide. So we said up here that pyruvate comes from alanine. It's a derivative of alanine. And so this is actually the step that is required in order to make alanine. And this is the step that's required to make valine. and leucine. So also knowing these steps are really important because it shows that amino acids can be made through transamination reactions. Okay, so now we're going to get into drawing pyrimidines and purines. So make sure you're familiar with the structure of a purine and a pyrimidine. Okay, so let's draw their skeleton. So in terms of like thymine, uracil, just be able to recognize those, but knowing how the skeletons look like are also really important, and knowing which ones are purines and pyrimidines are also important. A way I like to remember which ones are purines is pure as gold. So purines are the adenines and guanines. So if you can remember this, then you'll know that the other ones are pyrimidines. So purines have a 2-cyclic structure. So they look like this. And you can also find this in Dr. Thomas's slides as well. So this is a purine. Okay, and then a pyrimidine looks like this. I drew this really ugly. Okay. So they just look like that. And then just being familiar with recognizing how cytosine, uracil look like, and how uracil is found in RNA, thymine's found in DNA, stuff like that's really important. Okay, so let's move on to the pyrimidine pathway. So for the pyrimidine pathway, you begin with carbamoyl phosphate. Okay, so I'm going to draw the structure for this just so it's easier to explain. So there's 2 shared electrons, there's a phosphate in the middle. Okay. So, before we get to carbamoyl phosphate, there is a step that's needed to get to this process. But this is actually, once you form this carbamoyl phosphate, that is the rate determining step for the production of the pyrimidine. So let's try to go back and see how we can make this. So I'm going to just draw that in pink. So you start with bicarbonate. Okay, so you have shared electrons, okay, and it requires the input of ATP so we can transfer on that phosphate group. And you still have the structure which is similar to the bicarbonate here, but now you have the input of this phosphate from the ATP group. And this is called carboxyphosphate. And this is a very high energy bond. Okay. So because this is a high energy bond, that causes the next step to be irreversible. And you have NH3, which is added from glutamine. And you remove a phosphate, an inorganic phosphate, and glutamate. And you have this NH2. So you removed this entire group that's here. Okay? So this is called carbamic acid. Okay. And this next step is actually catalyzed by CPS2. So I'll draw what's needed. So you have ATP, and that gives us ADP since we're transferring a phosphate group. And this is catalyzed by CPS2, which stands for carbamyl phosphate synthase 2. And that produces this structure here, which is the carbamyl phosphate. So unlike the phosphate that was here, which was high energy, this phosphate here is not high energy. And that just has to do with the neighboring atoms that are surrounding it. So here you have a high acidic structure, and here you just have an amide. So it causes this to not be a high energy bond. Okay. So now that we have our carbamyl phosphate, we can start the whole pyrimidine pathway. Okay, so I'm just going to use this one that's right here. Okay, so the first step is a reversible step. We add aspartate, which is an alkaloid amino acid, and we remove an inorganic phosphate. The inorganic phosphate is the one that's highlighted here, and we form something that looks like this. So as you can see, the aspartate group, I'm not going to focus on dashes and wedges for this, just to also save time. I just did right there. But you get something that looks like this. And this is called carbamyl aspartate. Okay. And then from this, you have a condensation reaction. So you're adding in, condensation, removing water. So you remove water as you're adding in protons, and this is condensation. And you form this structure here. And what that condensation reaction allows us to do is form that cyclic ring. And this is called dihydroate. And then from that, you add NAD+, which gets reduced to NADH. And this is a redox reaction, so it's catalyzed by dehydrogenase. And as the NADP+ gets reduced, the dihydroate gets oxidized, and you form this structure here with a new oxidized part. And this is called orotate. Now, orotate is an intermediate. It is a precursor for all pyrimidines. So what happens is you would actually add orotate with a specific sugar with a phosphate. So, for example, I would have to add this ring. So that's showing that it's going in with something that looks like this. I'm not going to add the, actually I'll add the components of it just so it makes sense. And a linkage of phosphate groups. So something like this. So I would add this structure to the orotate, and this is also a reversible step. You would actually lose a pyrophosphate, and that pyrophosphate is coming from these two phosphates, so a pyrophosphate's two phosphates, and you would add the attachment. So you would get something that looks like this. Plus your pyrimidine ring. So this is the base. I'm just going to denote it as base, whatever base it is. Okay. And that's how you form your nucleoside. And this is also catalyzed. So this molecule right here is PRPP. And PRPP is synthesized from ribulose 5-phosphate and the addition of ATP, and that's actually how you create the structure here. Okay, so once you have this orotidylate structure, so this isn't the complete base. This is just like an intermediate base, so it's not specific to uracil or thymine. So you would actually add protons, which removes the CO2. So if you look in his slides, I'll just draw it here actually, so that way I don't confuse anyone. But it looks like something like this. So this is an intermediate step. So you're actually removing this CO2 here, so that way you can make it specific to your pyrimidine. And you would get so my structures are very ugly, but I have the right amount of carbons and elements needed, so I apologize for that. And now you have uridylate. So now this is a nucleotide that can act as a precursor for RNA. Okay. And then another concept that's important for this is understanding that UMP, right? So UMP only has one phosphate. But when you add it with ATP, you form UDP because you only add one phosphate at a time, and ADP. And this is through nucleoside diphosphate kinase.